Okay, good afternoon, everyone. In the last class, we were discussing about Western blotting and other techniques. We stopped at Western blotting. So in this class, we will discuss about a little bit extension to on the protein side. We have in the previous classes, we have learned about analyzing at the level of genes and variation. Then we learned about expression at the level of mRNA, that is transcriptomics. Now we started yesterday trying to analyze with at the level of proteins. That's where we came to Western blotting and stopped. And today's class we will focus a little more on that, and this will be followed by our general approach to finding gene function. Okay. So as we are all aware, there are several types of amino acids. Some of them are have a positive charge, some of them have a negative charge, some of them are neutral. Right? They are all amino acids. This is net charge we are talking about, net charge. But otherwise, all amino acids are amphipathic in a way that they have positive and negative charge. The amino group is the positive charge, and the carboxy or the acidic group COOH is the negative charge. But they may have additional negative or positive charges on their side chains. Based on that, the amino acids can be basic or acidic or neutral. So if a protein is composed of a large number of a protein may also have a net charge based on the composition of negative or positive charge, or acidic or basic. So every protein has a certain characteristics. If there are two proteins, they can differ in several ways. One is they can differ on the number of amino acids. They can differ in the sequence of amino acids. They can also differ, the composition also, right? What kind of proteins are involved? So if I have a situation where I need to know that there are two different types of cell types from the same organism, or one treated and one untreated, something like that, then I want to know what are the different types of proteins in A, and what are the other proteins expressed in B. I want to know all the proteins that are expressed in A and all the proteins that are expressed in B. And then that means we are actually trying to know the total proteins expressed. So we will call it as proteomics, right? So if you remember, as I was talking about, this is if you are trying to know about all the types of proteins specific to a cell type or a growth condition or a treated, untreated, something like that, all those things come under proteomics. So as I was talking about the amino acids and their charge, and because if they have negative or positive charge, and if you put it in an electric field, something like this, and they will move in either of these directions. If it is a positively charged amino acid, or here we'll talk it as if it is a positively charged protein. One second. Just hang on. I have a little bit of trouble. One second. I'll just be on. Yes. So I was trying to explain about the amino acids and so on. So what I'm trying to do now is that, one second, let me start the presentation. Yeah. So every protein, based on its composition, may have maybe either positive or negative, right, as a net charge. So that means, and also one more thing, I actually was trying to describe about amino acids and trying to tell you about the fields. And if you put an amino acid here, which is positive charged, it is likely to move towards the negative electrode. If it is a negative electrode, it is likely to move towards the positive. If you put a neutral one, it is unlikely to move anywhere. And different amino acids have different isoelectric point, in the sense that at different pH conditions, say if I take a low pH and here is a high pH, if I have a strip that has a different pH, and if I have so what is pH again? Right? When you say it's a negative logarithm of hydrogen ion concentration. When you say low pH, you actually have very high amount of hydrogen ions. When you say high pH, that is 14, you have less amount of H+. So if there is an amino acid which is a positive one, okay, I put it here. Positive, if I said it is NH, it has an additional amino group, NH2. Okay, I'm only drawing the additional one on the side chain. CONH is also here, okay? N terminal or NH2 and COH, that's common for all amino acids. Right, this is a common group, and on the side chain you keep actually having. So this amino acid has an additional NH2. So it might lose one of the protons and it might become positively charged. So what we should know is at certain pH, the positive charge will be neutralized and it will become net negative charge, net zero. And those are called as zwitterions, right? Those that have the net charge on that molecule is zero. You here, if it is neutral, it will not get attracted to either of these sides. Okay, I would suggest you and encourage you to look up. I hope in the last class I told you to have a look at that. If not, even now you should try. Just because the competitive exams are coming up, it will be a good idea to start with these. So at 1 pH, it becomes a net negative charge. So if it is going this way, you will have H+ added, and then it becomes a neutral one. Okay. Once it becomes, that is, it forms a zwitterion, it doesn't, it is no longer positive. Okay. So it becomes neutral, and because of which it will not, in that form, it has net zero, and because of which it will not move in either of the conditions. So amino acids are kind of amphiolites. You have different types of the different amino acids have different charges based on which also size matters, but it's not important here. Say, for example, in this, the technique called as isoelectric focusing. Isoelectric is about where the protein forms a zwitterion. Here we're taking proteins. We isolate the total proteins, and then we put it on in the middle of a strip. Strips come in different ranges, say 4 to 8, or some of them could be even broad range, say pH 2 to something else like that. So this is the pH. This is low pH. That means it should actually be having high amount of H plus ions, and high pH. It is alkaline conditions. You put proteins here based on their charge, and you also apply electric field, right? Based on charge, they will start moving in different directions. If it is a positive or basic protein, it will try to move towards the negative electrode. If it is an negatively charged protein, it will move towards the positive electrode. And then you have separation of different proteins. And this protein, for example, stopped moving further because at that pH, it must have become a zwitterion or net negative charge. Because of that, neither of this electric field that is applied is going to have any influence of it. So what is, why at this particular pH? That depends on the electric, the chains or the side groups that it has. Okay, so we can separate all these proteins based on charge alone. Okay, so in this case, you have one band formed. This band if there are several proteins with the same charge, they would have grouped and formed one band, right? Different types of proteins, even though they might have differences in their size, they will travel together because they share or they have similar charges. And then what you have to do is you put this strip here and you perform STS page, the one that we generally do in the labs. Now, STS page is you have sodium didecyl page gel. So it is a denaturing gel. All the proteins are now singled out and they are stretched out in a way. Sorry. And then all the proteins will have a net negative charge. So charge doesn't have any more role in it. And now from each band, say for example, in this band, there are 3 different proteins. Based on size, they will move like this. If this one had 2 proteins, it may move something like this, right? So eventually you will have a gel showing points something like this. So this is referred to as 2D protein gel electrophoresis. Why is it 2-dimensional? On one dimension is isoelectric focusing. We are separating the proteins based on charge alone. On the second dimension, we are separating based on size. Because charge doesn't have any more role because we have used sodium didecyl sulfate. So now they have all become negatively charged, and then they would move only based on their molecular size. The larger one will travel slow, the smaller one will travel faster. So I hope you understood the 2-dimensional, and it is protein gel electrophoresis. Why do we use that? This will give us the total types of proteins. Usually what you can do is take this protein and you can cut it out of the gel and characterize. You can send it for sequencing, protein sequencing. Nowadays we don't use that anymore, but in the olden days that is also a possibility. But to cut out and extract from this small band protein, if you want to sequence protein, it is possible, but you need a large amount of the sample. Okay, so otherwise the revolutionary thing about proteomics is mass spectrometry. We can compare different types of organisms and perform two-dimensional gel electrophoresis. Say, for example, I performed two-dimensional gel electrophoresis of sample A and sample B. say if I am having a dimensional something like this, so the common bands, if there are common, that means they are expressed in both the cells. If one is present in something like this is one is present in one type and it's absent in another one, then this is specific to cell type A or sample A. So you can segregate those and you can find out which one is the differences between two organisms or treated, untreated, and so on. So typically you would extract the protein from one of the samples, whichever you want to. If here, assume that you're comparing two different types of plants. So you take the protein from leaves, assume that, or some other roots. Same sample you would use from the other one, other plant as well. And you perform 2D gel. This is how it would look like. It doesn't look like clear cut bands. That's one of the difficulties that we will have. And you can perform 2D electrophoresis of two and overlay them, and computers can actually perform a large image analysis to figure out which ones are specific to sample A and which ones are specific to sample B, and so on. So then you can isolate, you can figure out which ones are the differentially expressed proteins. You can select them, and that spot is cut out. You can cut that part of the gel, and then you use trypsin. It's a protease. It cuts the protein after arginine, I think. So it will cut, you have a larger protein, it will cut into smaller fragments. And these peptides from that protein are fed into mass spectrometry. Mass spectrometry by itself is a whole course, actually. I'm not going into the details. It will be difficult. But you have something called as, I hope you learned these in analytical techniques. MALDI, electron spray ionization. There are two different techniques predominantly used in proteomics. This is matrix assisted laser desorption ionization, and this one is electron spray ionization. So in MALDI, it is put onto a slide with a kind of material, and these peptides are put, and you hit it with a laser, because of which the peptides might become ionized, and they will move into the tube. So there are, in spectrometry, there are two parts. One is about ionization, and the other one is an analyzer. Everything is automated. We only sample preparation is made and put here. If it is MALDI, laser will hit and the sample will go. Analyzers can be two types. One of it is time of flight, and the other one is quadrupole. You don't have to know all these things as a part of rDNA, but if you are interested, there are a lot of YouTube videos which will help your understanding much further. So time of flight is a simple box, at least that I am very clear of. So the ions will move and will hit this place and will start moving back. And here you have the detector. The larger ones will travel faster. Smaller ones will travel, larger ones will travel slower, and faster ones will travel faster, smaller ones fast. So that is time of flight. Based on that, you have ionized here, and all those proteins peptides are based on the time of flight. The computer can generate a cardiogram, something like this, which will tell you at what peak what is coming out as peaks like this, and there will be a lot of noise. One needs technical expertise to figure out what is a peak and what is not. And there are also a lot of corrections that have to be done to the peak, the electrogram that is generated. And then you can compare it with theoretically generated fragment sizes, because you're using trypsin and it is cutting only at specific locations. And we have a large database of genomes and proteomes. We know about almost all types of proteins that we are working or around us, majority of them, if not all. You can compare the molecular weights that the mass spectrometry is showing with that of theoretically generated peptides from different proteins from different various diverse organisms. So it would hit into one of the same. I mean, either the molecular weight generated like this and theoretical molecular weight, there will be a close match, and that you will take it as the protein identity of the protein. Okay, so proteomics is a very interesting and powerful technique. It is used many places, including diagnosis and in cancer study and so many other. It is there in everywhere. So I think that is about proteomics. Proteomics is the study of the total proteins expressed in a cell type, which can be used for comparison with other types of organisms or other types of cells. So electron spray ionization is also simple. I'll just use this part. You will put it into a syringe-like thing, the sample, and the sample you would have applied a huge electric field here, I mean, to the needle and somewhere. So whatever is coming will get sprayed into like this and becomes tiny droplets and burst and becomes ionized. The peptides become ionized, and they are then detected by any of the time of flight or quadrupole analyzer. I'm not very familiar with, or I don't remember all the details about physics of the quadrupole analyzer. But the point is, the machine mass spectrometry will be able to measure the mass over charge of all the peptides that are passing through the analyzer. You are ionizing, and then the analysis is based on what charge does it have and what mass does it have. M over Z is given, and then that can be compared with the already existing one that is called as peptide fingerprinting. Okay, it's worth remembering that, I think. Peptide fingerprinting, and then you will have identified the proteins that are specific to either of the cell types or samples that you are analyzing. Okay, now we are going to move into a methodology. Okay, method is different from methodology. Method is the kind of technique or an essay that you're doing. Methodology is about the logic or sequence of techniques and why you are using that sequence of techniques is methodology. All those come under methodology. There are two phrases that are used, forward genetics and reverse genetics. Forward genetics is if you know about, say, for example, skin color, okay? And you are observing a trait or a phenotype, and then you're trying to find out what is the gene that is causing this phenotype. That's traditionally, that is what people see. They didn't have information about the gene, right? They only know about the phenotypes. Now, in the, say, last 100 years or so, we are much more aware of genes. So if phenotypes, people observe phenotypes and then they try to find out the gene. So it is forward genetics. With once the genomics and the genetics, genes, DNA, structure of DNA and other things are done, so many people have sequenced a lot of organisms, and now we know a lot of genes, but we don't know what they do in the cell. So we know the gene sequence, but we don't know the phenotype. That would be something like reverse genetics. Okay, so these are two different methodologies or approaches people use, and For that, they can construct genetic screens, which we might not discuss those in detail, but in one of the papers, we will have using CRISPR and genetic screen. I think it is there. So maybe we will discuss at that time. I hope you understood forward genetics and reverse genetics. From phenotype to the underlying the gene, if you are trying to find the gene that causes the phenotype, it is forward genetics. If you know a gene and you're trying to find out the trait or the phenotype, then it is reverse genetics. So there are several ways of the approach. What people do is you will find in reverse genetics, in forward genetics, typically we will try to find mutants, and we will see which gene is mutated. Mutation. We will take an organism. We will try to generate a mutant. Okay, say this is a mutant cell. Now it had this round phenotype, and it is no longer round. This is a mutant. So what caused this mutant is what we are supposed to find. What genetic element caused the loss of the round phenotype is one of the approaches, isn't it? If you are taking in terms of reverse genetics, you take a cell and you have a gene, you don't know what it is doing, so you put it into it and you overexpress it and see what happens. Okay, then because this gene is added and or overexpressed, you find a phenotype. Then you associate the genetic element with that of the observed phenotype. Okay, so reverse genetic is much more difficult in general, but it's possible. Both are difficult. We need to have a lot of theoretical understanding and thinking and using the principles appropriately. So here is one of it based on mutagenesis. So I will try to make up one of it. Is the objective, for example, is that I have a cell, and it has it can, when I add X-gal to this in the presence of the cell, it gives blue color. Okay, that is the phenotype. I know the phenotype. I don't know which gene is causing it and where it is located. My objective is to find all the genes that are required for it, so I can generate mutants. You know about transposons, TN, and here is you put a transposon on a temperature sensitive plasmid, and I infect this cell with such kind of transposon. So transposon, what will it do? It can transpose from the plasmid to some part in the gene. I hope you remember the phrase insertional inactivation, right? If the transposon got inserted into a gene and it did not lose the ability to give rise to blue color, then wherever it might have integrated, the gene is not responsible for generation of the blue color. Say, for example, in a different cell, transposon has insertionally inactivated gene X, because of which this particular cell can no longer turn X-gal into a blue colored compound. Then that means gene X is involved in the, directly or indirectly, involved in the conversion of X-gal, a colorless one, into blue colored compound, isn't it? That's the approach. We know the trait. This is the trait, and we are trying to find the genes responsible for this trait, and we are making an approach. We're taking an approach by using or by creating mutations in because of which the phenotype is lost, and then we can find out where that mutation is. Okay, let's see how we can do it. So I hope you understood this. You take this plasmid which has the transposon, you have to transform it, and then you will allow the transformants, and then you change it to restrictive temperature. Here it is taken is temperature sensitive plasmid. So it would mean that the plasmid would replicate at 30 degrees centigrade, but would not replicate at 42 degrees centigrade. Okay. So if you have a cell and the cell has plasmid and it is at 30 degrees centigrade, every time the cell divides, what will happen? Plasmid also will replicate along with it, right? But if you change this cell to 42 degrees centigrade, the plasmid cannot replicate in that situation. So it will not come to the progeny. One cell may inherit the already existing one. The other cell is cured of the plasmid, right? That is how one uses temperature-sensitive plasmids. Okay? So you have transformed this plasmid containing transposon. Then you allowed transform in the permissive that could be around 30 degrees centigrade. Then you change to 42 degrees centigrade. Now the cells that lost the plasmid will get selected. This transposon also has an antibiotic resistance marker. Okay, so at 42, majority of them are without plasmid, right? And if they are still able to grow on a resistance, something like say ampicillin, what does that mean? If they're able to grow on ampicillin, that would primarily mean that they are transformants, right? This is where, because the resistance gene is on the transposon, which is on the plasmid. But after transformation, you put it on restrictive temperature. So at 42 degrees, the plasmid will no longer replicate. And so because of which you will have a large number of cells that are cured of the plasmid. There is no plasmid, right? And still, if they're able to grow, that is possible only if the transposon, which was on the plasmid, okay, assume this is a plasmid, has jumped onto the genome, right? Suppose something like this happened. Assume that you have grown this for several generations or overnight. So only one of the cell will have the plasmid. All the rest of the cells, if they are growing, they were growing because the transposon has moved on to the genome. So in this method, in this step, what you're doing is you are selecting for all the mutants, all the cells that have got the transposon on the genome. Okay, so this is what you would actually find, because we have stopped the plasmid replication, but the cell division is happening. So all the new cells that would form would actually not get the plasmid. But if they are yet still able to grow, if there are anything that is able to grow, they are growing only because the transposon has hopped onto the genome, and the transposon has the antibiotic resistance gene that is shown, that I indicated here. Okay. Now you found out you constructed a huge transposon mutant library. Transposon would move into any one place. You don't know where it will move. It is a random integration process. And also one more thing you need to remember is the transposase gene is present on the plasmid outside the transposon. This is the transposase. So if the plasmid has, sorry, I think, okay, if you didn't understand, it is all right. I will try to repeat in the next class. Right now, try to remember, try to follow a little bit. The screening would usually require 2 times of explanation probably. So what you have now done is you have a large number of cells, and assume this is the genome. You have A, B, C, and D. Those are the genes. So in one cell, this is where the transposon is hit. In another one, it is hit on the B. In another one, it is hit on the C. In another one, on the D. Sorry, yeah. I hope you are able to follow what I'm trying to say now. So we have a mutant library. The genome has 4 genes, and in each different cell, you have one transposon. They indicated with the red dot like this. And we are going to screen for the loss of a trait. Assume I will follow with what I was telling, the ability to make X-gal to a blue colored compound. I will see that which one has lost this ability. Assume that this is the cell that lost the ability. That means gene C is directly or indirectly involved in this particular phenotype. Okay, and now for convenience, I already wrote these are ABCD. How would I know that? This is reverse genetics. We know the phenotype. We know nothing about the genes or the gene sequence. I should be able to cut out the clone fragment and then do the characterization. Okay, we will come to that in a few slides. I hope you remember lac-operon. This is lac-I. It produces repressor. It is a little farther away. And you have the promoter operator region to which the polymerase binds. There are 3 genes, LAC, Z, Y, A. Z is beta-galactosidase, Y permease, and LAC-A is transacetylase. So if we have, for blue color formation, you need beta-galactosidase is the enzyme that makes X-gal2, which will convert X-gal into blue color. It is lactose operon, you know that. So what are all the essential things required for beta-galactosidase, this trait? We first need this enzyme. If this enzyme should be there, mRNA should have been produced, so you need the promoter. That means transcription should have happened. We need a functional one, so the gene should have been functional. So there are 2 essential elements in this case, that we need the expression of this gene and we need a functional gene, right? Only then you would have this X-gal converted into a blue color compound. So in the previous one, we made the mutant library and we want to know where is it. We don't know the gene sequence, right? It is a little bit, in all of it, I think I said you have A, B, C, and D. That's what I must have said. And B, in this gene, you have the transposition. So there is a nice technique called as inverse PCR. Please try to remember what is inverse PCR. Today's focus is only on inverse PCR. I hope you understood the transposition one. If not, maybe in the next class, I'll try to review it. Let me see. So assume that this transposon is hit on C, and I chose the cell which has lost the trait, a mutant. Okay, so what we do is, remember that we know the whole sequence of the transposon. Okay, so you can treat it with one of the restriction enzymes, but that restriction enzyme should not cut in the transposon. That's one of the limitations that you have. Then what you will get, you will choose a restriction enzyme that doesn't cut the transposon, and you'll take the genomic DNA and use that restriction site, say BAMH1. Okay, BAMH1 is not present site is not present within the transposon coding region or the transposon genetic element. Once you have taken this, what you're going to do is you're ligating. So this end is BAMH1 overhang. This end is BAMH1 overhang. This is the genomic DNA, which should actually be the fragments or the parts of gene C, right? Because that is the mutant we are analyzing. Now you add, when you add ligation, you will form a product something like this. It will circularize. Now you're going to, you can use a restriction digestion. Now you'll choose a restriction digestion that is present only in the transposon. Say, for example, EcoR1. So EcoR1, at least you know you're choosing EcoR1 so that the transposon will get cleaved. Now, if it is cleaved here and stretch it out, what will you get? You will get this was the parts of C, the gene C here, and these were the parts of the transposon, right? Previously, what is the situation here? If you look at here, you had the transposon like this, and the fragments of gene C like this. In between, what did you do? You have ligated, so it became circular, and then you cut it here. Okay, you joined these two overhangs, and you cut it here. Now, what was outside is now inside, and the transposon fragments are here. And that is one of the reasons why you call it inverse PCR. You are literally inverting this whole fragment like this into this one, where the outside fragments are going in, the inside ones are coming out, right? This is the way it is happening, isn't it? So, and then because we know the sequence of the transposon, we would have designed some primers, something like this, and then we would amplify. So we will get the fragment, which we will sequence. And then we will be able to find out what is that gene. I hope I hope you see that it is fantastic idea to start with. You can go back to where we started. This is where we started. The objective, I'm trying to review what I have discussed, okay? Try to follow and see. For this to happen, what all do I need in the transposon? Now we have the bigger picture, so let's see it whole. I'll try to describe the vector first. The vector has origin of replication, which is temperature sensitive. The transposon also has an antibiotic resistance gene, say AMPAR. The blue one, this is the transposon. So that is the whole thing is transposon. We know the sequence of it. The plasmid has the transposase gene also. We are not having the transposon with the transposase gene, because at one time it is like here, in another time it might jump to some other location. That will complicate our analysis. So we are putting it on the plasmid. So this should have produced the transposase enzyme that will recognize the ends of the inverted repeats at the ends of the transposase, will cut and will move this from here to some other place. For selection of or generation of the transposon mutant library, we would have transformed this plasmid into a large number of cells. Then we would allow it to grow at permissive temperature. Permissive would have been 30 degrees centigrade, where we are allowing the replication of the plasmid, so that you'll have a large number of cells with the plasmid. Then we are shifting it to restricted temperature, that is about say 42 degrees centigrade. The cells will grow, but the plasmid does not replicate anymore, because of which majority of the cells will get cured of the plasmid. And then we are placing it on ampicillin plates. So ampar. So those the cells that they will not have plasmid, because we are growing it at 42 degrees centigrade. They are cured of plasmids. And then they're able to grow on ampicillin because they're having ampicillin resistance. They can grow only if they have got this transposon onto the genome. Okay. And that is how one would generate a large transposon mutant library. And you will check for in the mutants, which all have generated or lost the phenotype. You are looking at mutants. Now we know that transposon has hit onto a genome, which is inactivated, chromosomal insertional inactivation, and that is why we lost the phenotype. And now we need to find out what is this gene sequence, or what gene is it. For that, you need a technique like inverse PCR is fantastic to use. Here is the transposon that is hit onto a gene, assume from here to here, and this part is the gene. But because transposon is integrated, it is insertionally inactivated. Now you will also digest it with several with a restriction enzyme. That restriction enzyme should not, the choice of the restriction site here, restriction digestion in the first step, it would be based on that should not have been, that should not cut the transposon. So we will choose a restriction enzyme that cannot cut the transposon. And then now we will get fragments with the transposon. There will also be fragments without the transposon. We will also have them for comparison. All of them, assume we have used BAMH1 here. So all the ends, overhangs are BAMH1. And now we use ligation. So there are many possibilities, out of which one of the possibilities is that you'll also have circularization. Okay, this is the transposon. For these, you'll also have the molecule circularized like this, but there is no transposon in them. Now we will digest it with a restriction endonuclease, say EcoR1. And it will cut. We are choosing EcoR1 or other restriction endonuclease based on unique site on the transposon. Okay. So these also may be cut if it is EcoR1 is present. These also may be cut, and sometimes you should choose. There is a little more detailed protocol, but general scheme is this much is fine for understanding sake. And then we would have, once we cut out, we would have inverted the fragment. The transposon sites are on the outside, and the gene fragments are in the inside. And because we know the sequence of the transposon, we would have used transposon specific primers for amplification, followed by sequencing. And that is how we can figure out the gene responsible for that particular trait. Okay. Yes. Any other questions? For simplicity, I will add one I will add a one step that is there. Usually, which site should you use a 8-base cutter, 4-base cutter, or 6-base cutter when you're using in this step? Is the question. I want frequent cutting. right? Because it's a genome, it's huge. So typically a 4-base cutter is better to use in that time. That 4-base cutter sequence should not have been in the transposon site. That's the one precaution one people should take. So because of which you will have much smaller fragments here formed, as opposed to if you used a 8-base cutter, you will have a large one. If you had these large sequences, then in the second restriction site when you're using, it might, after circularization, you will use the second restriction enzyme. In that case, this part also may get cut, right? And then we will not have a product formed like this. So typically when you're doing in the first place itself, you should have chosen a restriction site that is shorter in sequence. And I will also add one more complication like GATC, GGATC, sorry, CCTAGG. So this is the sequence for South3A. And this is the whole of it is sequence for, say, BAMH1. Isn't it? Okay, I'll finish in last 1 minute. So I would use here, I will use South3A. And here I would use BAMH1. Why am I doing that? Say I've changed the restriction endonucleases. So now you can try to tell me. You have to think in that way. All BAMH1 sites will be cut by South3A. But not all BAMH1 sites will be cut by South3A. But the reverse is not true, not necessarily true, isn't it? South3A only recognizes the middle GATC, isn't it? So all BAMH1 sites will have this GATC. So all BAMH1 sites will be cut by South3A. Whereas all South3A sites don't have to be cut by BAMH1. They will only be cut if you have the G on the 5' side and this C on the 3' side. Otherwise, BAMH1 cannot cut South3A sites. So that's how you have to think. So if we have used South3A, that means the fragment that is formed here does not have any BAMH1 sites. Do you agree with that? If I already used here South3A, this part of the gene, the genomic DNA, does not have any BAMH1 sites. If it were there, South3A would have cut it already. Okay? Think about it. And if you have any questions, we can again discuss on this in the later class. Okay? But any questions, please ask. Okay, watch the video and try to come up with questions. This is complicated. Try to watch and then we can have a discussion in the later class. Is that fine? Okay, take care then. We will meet in the next class.